Good morning, everyone. Welcome to Research Radar webinar for April. A lot of prompt attendees today. So while we're still waiting for folks to hop on, if you can put in the chat box what your name is and where you work so we can have an understanding of who's on the call, that would be great. I am seeing a lot of folks from MnDOT, Hennepin County, St. Louis County, Illinois. Welcome, Amber. More MnDOT folks, DPS, Minnesota Valley. Fantastic. So it looks like we have quite a few people already joined on. My name is Michaela Kranz. If you are just joining, if you can add to the chat box what your name is and where you work, just so we can know who is on the call, that would be great. So we're going to go ahead and get started. Thank you for joining today's Research Radar webinar, Environmental Innovations, hosted by the Minnesota Department of Transportation and the Minnesota Local Road Research Board. My name is Michaela Kranz. I am the Research and Innovation Marketing and Communications Manager. We're excited to have our presenters Jesse Schmidt, Ryan Foley, and Chris Smith joining us today. So today's webinar is the fifth in our new Research Radar series sponsored by the Minnesota Department of Transportation's Office of Research and Innovation and the Local Road Research Board. Each month, we're going to highlight 3 research projects focused on a common theme. So last month, we featured community livability research projects, and we'll share a link to that recording if you weren't able to attend. MnDOT's Office of Research and Innovation manages more than 185 research projects in progress, ranging from local initiatives to pooled fund projects with other states, comprising both basic and applied research. So we're here to help transportation practitioners determine whether new research is needed or if existing studies may meet your needs, recommend the best research methodology for your project, access local and national funding sources, and then manage your project from start to finish. The Local Road Research Board is a city and county run organization that sponsors research and educational initiatives that address local transportation needs. So you can stay up to date with our research by following us online and subscribing to our newsletters. We'll send you a link to the website and subscription links in a follow-up email after the webinar. Our research database you can see on the screen looks like that. We're also excited to announce the funding of 27 new academic research projects selected from a competitive pool of 82 proposals. To see the full list of new projects, you can scan the QR code on this screen. And if you have subject matter expertise on any of these projects and are interested in serving on a technical advisory panel, please reach out to our office. So before we get started, I'm going to read the Tennyson warning. This session is going to be recorded for those who can't make it. If your camera is on, that might be captured in the recording. You are not obligated to show your visual. So if you would prefer not to, just turn your camera off. It's as easy as that. And now moving on to the good stuff. Our presenters for today. So presenting on identifying deer vehicle collision concentrations in Minnesota, we have Jesse Schmidt. Jesse joined MnDOT's Office of Environmental Stewardship in February 2024. She has a master's degree in conservation biology and over 9 years of experience in natural resource consulting. Moving on to our second project, Species from Feces, a new tool to identify bats in culverts and on bridges. Ryan Foley has been with MnDOT for 5 years and works in the office. I see a little whoop whoop. Has been in the Office of Environmental Stewardship, Environmental Assessment Unit. Before coming to MnDOT, Ryan worked at a consulting firm conducting freshwater mussel and fish surveys throughout the country. He has an undergraduate degree from the University of Minnesota and a master's degree from Tennessee Tech University. And our last project is a new tool for early detection of endangered turtles on proposed transportation projects. Chris Smith has been with MnDOT for just over 9 years and works with the Office of Environmental Stewardship Environmental Assessment Unit. Chris is a wildlife biologist by training and has expertise working with amphibians, reptiles, bats, and insects. So before I hand it over to Jesse for our first presentation, it is an International Appreciation Day of one of these three animals. So if you have your guess on which of these three animals, deer, bats, or turtles, is the Appreciation Day, put that in the chat. If you Google the answer, we won't know, but you will know that you Googled it. Okay. Seeing your guesses come in, we're going to reveal it later on. Okay, I'm going to hand it over to Jesse for our first presentation.
All right, thanks, Michaela. I'm going to go ahead and share my screen. Let me know if you can't see it or if you have trouble hearing me. But I'm going to assume it looks good. Oh, wait, present. Okay, here we go. All right. So, thanks everyone for coming today. You may be surprised to learn that the deer is actually the deadliest wild animal in the US. So I asked my kids this question, which animal they thought was the deadliest. And my son said rattlesnakes, and my daughter said sharks. So they were wrong, but they were in line with what a lot of other people might think of when it comes to a deadly animal. You might think of wasps or bees or snakes or maybe mosquitoes or ticks. But no, in fact, it's just the sweet little deer. And it's actually not too surprising when you think about it, because if you drive regularly, you've probably hit a deer. And if you haven't hit a deer yet, you've probably seen a deer carcass on the side of the road. So it's extremely common. And when you hit a deer, they're big enough to cause substantial damage. So in Minnesota, there are up to as many as 6 fatalities per year caused by deer vehicle collisions or DVCs. And so not only are they a safety hazard, but they're also economically quite costly. So the estimated average cost of a single deer vehicle collision in Minnesota, I believe, is around $13,000. And that not only includes the market-based value, which would be like the vehicle or property damage, medical bills, and things like that, but also there's a passive use component as well, which is the non-market or the intrinsic value that's lost as a result of that crash. And so there are it's an average of about 1,200 DVCs reported annually in Minnesota, but it's suspected that many go unreported. And insurance companies actually have their own estimates that are in the range of about 35 to 40,000 collisions annually. So it's much higher than what's reported. And so if you take those estimates, those higher estimates, and multiply that out by the average cost, that's an estimated total loss of about $500 million. And so if we could just try to reduce this number by even a small percentage, it would make a big difference. And so that was kind of the impetus behind this research, because it's pretty impractical and super expensive to fence the entire roadway. You can't do it all everywhere. MnDOT was interested in identifying high-risk areas for DVCs on Minnesota roads so that we can prioritize accordingly and target those hot spots where deer vehicle collisions are occurring. And so there were kind of, this research was conducted by the University of Minnesota, Duluth and Twin Cities, and in partnership with MnDOT. And they kind of broke it down into 3 different steps. So first, identifying the relevant variables in deer vehicle collisions, road characteristics, and things of that nature. And then they did a field component to estimate the reporting rate in Minnesota. And then finally, to use all this information to create a model to actually identify where those DVC hotspots are occurring on our roadways. So first, the literature review. There are a number of different variables that go into this research, many being road related, and there's a wide number of variables studied with this. But the most common would be traffic volume, or average annual daily traffic, AADT, and traffic speed. So that makes sense. The more cars on the road, the more likely you are to hit a deer. Other variables included road width, number of lanes, lighting, road density, road curvature, which would impact the sight distance. And these studies have been conducted across the country, and many of them had different and contradictory findings. So there weren't too many conclusive factors, but traffic volume and traffic speed were big ones. Then there's also the deer-related factors. So you're more likely to hit a deer during those times when the deer are most active. So that would be deer behavior, not only their daily activity patterns, but also their seasonal patterns. So throughout the day, they're more active just before sunrise and just after sunset. And so you're more likely to meet them on the roadway then. But then seasonally, you can see in this graph here, this is from reported collisions in Minnesota. There's a small peak in June, and that's during the fawning season when little Bambi is more likely to wander onto the roadway. And then there's a big peak in November, and that coincides with the breeding season when the deer are just way more active and not paying attention to their surroundings. And that also coincides with hunting season as well. Then another deer-related factor would be deer density. So logically, more deer equals more crashes with deer. And also habitat-related variables, primarily the land cover. So what kind of land cover is around the roadway? Is it forested, grassland, developed, open water? These all factor in, and also the configuration of those land cover types. So there's a lot of different variables, and it gets complicated pretty quickly. So this research was trying to distill it down into a pattern. And so the next portion of the research was the field study, in which the researchers drove selected routes primarily around the Duluth area, but then they also did an extended search outside of that for a few months to collect actual deer vehicle collision data. And so they would just drive over or around 1,000 miles per month. And if they saw a deer on the side of the road, they would pull over and collect a GPS point and some pictures and other notes to try to estimate the time of the collision and other information that they could use in the modeling. And so they took all these data points of collisions and they compared it against the Department of Public Safety's data and used that to try to estimate the reporting rate of DVCs for these road segments. So they broke the roads into 10-mile segments to do this analysis. And the reporting rate did vary quite a bit, from 0 to 38% on these roads, but the average was just around 10%, so very low. And actually, they found that in about the past 20 years, there's just been an overall decline with reporting these crashes. So it is a much bigger problem than what's reported. So then they took all of this data, the crashes, and then also all the road characteristics that they had available, and put them into numerous statistical models to try to explain the locations of these crashes, to figure out what factors were the most significant. And so this table just shows the results of the linear regression model, which showed that the traffic volume and travel width and deer density all had a positive effect on the deer vehicle collision rate. So meaning if you have high traffic volume and a wider road and more deer, you're more likely to be in a deer vehicle collision hotspot. Then they also included winding factor, which is the road curvature, number of culverts, and number of stream crossings. Those had a negative effect on the likelihood of a DVC. So possibly more stream crossings and culverts. Those are opportunities for the deer to get across the road without having to get on the road. But the most significant variables were the traffic volume and deer density. So more cars and more deer equals more crashes. So then they used one of their models to calculate a DVC index for these road segments so that they could apply it across a map and visualize these hotspots. So a DVC of 0 would be equivalent to, no risk of a DVC, versus a 10 would be the highest risk of DVC. And so as you can see on the statewide map, the red roads are the top 15% riskiest road segments across the state. So there's really a big red blob in the Twin Cities metro area, and kind of following the main highways out of there. But then you can also see, unfortunately, we have these gaps. Those gray roadways are where there wasn't enough data to run the model, so we don't have the information for those areas. And so next steps with this research would be to, we have the statewide map already, but to create a set of district-specific maps so that individual MnDOT construction districts can identify the top high-risk roadways in their areas. For primarily, it would be useful for project scoping, so that if there's a project going on and they cross-check it against their map and they can see they're in a hotspot, then they might be, that would influence the types of prevention measures that they could implement. So a good example of this would be a bridge replacement project, where they're trying to decide what type of structure they want to use. And knowing that it's in a DVC hotspot, they might be more inclined to replace that bridge with a span bridge as opposed to a box culvert, to allow freer movement under the roadway, or just to simply provide fencing as well along those high-risk segments to keep the deer off the roads. And so here's an example map that I made for District 2, and in the purple it shows the top 1% riskiest roads in that district. So I'm planning to make these for each of the districts and have that available as a web map in Portal, most likely. And then a next logical step would be to also address those gaps in the mapping. So again, those gray roads where we didn't have enough data to calculate the DVC index. I think it's primarily deer density data that we need to be able to run the model. And then assuming that these maps are useful, it would be great to continuously update them as we get more data available so that they're always current. And that's all I have. So I will turn it over to Ryan.
Yeah, thanks, Jesse. Great job. I would have thought sharks too, definitely, especially in Minnesota. See if I can figure this out. After 5 years of using Teams and sharing screens, I'm still not getting it. One of these days. Is there like a present? Here we go. All right, how's it looking?
Looks good.
I guess I can skip forward. All right. So species from feces. The overall goal is whose poo is that? We've got 8 bat species known to occur in Minnesota, 2 of which are either federally endangered or proposed for listing. Bats use bridges and structures as roosting sites, whether it's an occasional night roost while they're kind of taking a rest on, while they're feeding, or if they're kind of utilizing bridge joints for their maternity colonies. Therefore, our bridge projects, whether it's a joint replacement, approach panel work, bridge replacement, these activities have the potential to impact endangered bats. The big issue is that oftentimes the bats are not actually present. They might occasionally use that structure as a rest stop during the night, but it's still kind of considered suitable habitat because they're utilizing the structure. But what they do leave behind are pleasant little turds, guano. So how do we figure out? Here we are at this bridge. We have to discuss this project with the Fish and Wildlife Service. What bats are using the bridge. Often we just have a pile of guano to go off of. So we kind of partnered with the University of Minnesota Duluth and the University of Minnesota to conduct this project to kind of determine, how do we determine which bat species are using the structure, and how to develop a statistically defensible sampling protocol that we might be able to utilize in our Endangered Species Act consultation with the Fish and Wildlife Service. So there's a lot of steps with this project. I've always been told, keep the number of words on a slide to the minimum. Obviously, I've ignored that advice. Step 1 is just kind of a literature review. What's been done previously. We don't need to reinvent the wheel here. That's been completed. Step 2 was the researchers kind of taking a look at our bridge database, seeing what kind of structures are out there. What's the characteristics of these structures? Are these, iron structures? Are these concrete structures? There's different, designs of bridges, or multiple spans, what have you. What kind of structures might have a higher probability of bat use? So you can kind of look at our bridge inspection reports, and there is a element on those reports where inspectors can say, like, yes, there's nesting birds on this bridge, or there's evidence of bats. Step 3 would be just collecting the poo. And the researchers did go out there this past year and started their collections, and they will continue with these collections this upcoming summer. Acoustic data collection, again, completed this summer and will continue this upcoming summer. The kind of idea here is, let's say you're collecting poo and you analyze through, genetic stuff, way above my pay grade. We see 3 species of bat based on these genetic analyses. Well, then you can kind of cross-reference with acoustic data collection and say, well, the acoustic detectors detected 6 species of bat. So, what does that say about our guano sampling protocol? Do we need to increase the sample size, the number of guano pellets to, detect those additional 3 species? The genetic analysis is still ongoing, in progress, no results as of yet. I guess I should mention at this point that this, the deer vehicle collision project is complete. This project is still in progress, so a lot of these results are going to be kind of, preliminary. After all the field work, after all the guano collection, after all the acoustic data collection, there's going to be some modeling involved to kind of answer that question of how many guano pellets might need to be collected. Do different species of bats utilize different parts of the bridge? So, spatial distribution of those guano pellet collections. And then step 7, the end result. The point of this is for us to be able to come up with kind of a defensible protocol that we can present to the Fish and Wildlife Service and say, hey, we've got this bridge project. There's a few, we didn't see any bats. We can't positively ID based on, pictures or direct observation. But based on this defensible protocol of the guano pellets, we have a, 90% probability that, an endangered bat is not using the bridge. And therefore, Endangered Species Act consultation goes a lot smoother. And then that diagram is, I guess, generally how DNA analyses works with this sort of thing. Do not ask me any questions about specifics, please. So what did the researchers find thus far? The bridge database analysis was complete. Bats were found on 26 different bridge types across, different materials, design, what have you. Almost all bridges that do have evidence of bat use are made of concrete. We believe that's due to, I think Chris or maybe somebody else came up with this, but it's called thermal inertia, which I really enjoy that term. Basically, the bats use the bridges at night to stay warm. 90% of the bridges that have evidence of bat use span waterways, which makes sense. Bats, feed on a lot of insects, maybe emerging aquatic insects. There's the river. This is great. It's one-stop shop. And the longer the bridge, the higher probability the bridge is being used by bats. And that may be just due to the sheer number of available roosting sites with the longer structure. Longer structure means more joints, more nooks and crannies, like a big English muffin. Guano collection was collected from 92 different bridges throughout the state, and DNA analyses are still in progress. They deployed acoustic detectors at 54 bridges, and the researchers detected 8 different species, including the federally endangered northern long-eared bat and the proposed tricolored bat. However, detections of these 2 species were uncommon, as you might expect, given that they are either listed or proposed to be listed. And there are some initial apparent differences in how different species utilize bridges. For instance, there is some some evidence that some species might selectively use bridges compared to nearby forested sites as kind of like a I don't know a environmental baseline. So their use is well, maybe not significantly. That's a statistical term that I'm not going to use right now, but some evidence that that may be occurring. And so the graphs on the right here, kind of hard to follow. But the top one generally shows, cumulative frequency distribution. For example, 60% of the deployments have 0 calls detected per night. 10% of deployments can have more than 200 calls per night. The bottom graph shows kind of for one species, kind of a nightly activity pattern. Kind of acoustic detectors, picked up surges in, calls right after sunset and just before sunrise. And for this particular species, calls were more frequently detected at bridge deployments than at forested deployments. So begs the question, are we actually creating habitat for bats with our bridge projects? So what does this all mean? At the end of the day, after all this fun research, what does it all mean? So the goal, as I've stated before, is kind of develop a sampling protocol with like a standard methodology where we can present guano analysis to the Fish and Wildlife Service to streamline our Endangered Species Act consultation. There's still a lot, to be determined. The researchers still have to run the modeling, the statistical analyses. We need to figure out, how many guano pellets might need to be collected on a given bridge. Does the type of bridge affect the number of, number of pellets and distribution of the pellets? For example, if it's a long bridge spanning, say, the Mississippi River, there's a lot of nooks and crannies on that bridge. Might that bridge need more sample size than a small bridge spanning, a railroad track, that sort of thing. But we're pretty confident that this is going to be a useful tool in our toolbox to help streamline Endangered Species Act consultation and make sure our project managers get off our backs about getting our consultation completed on time. And in these pictures, you can see Jesse there collecting guano samples from under the bridge. You can see that we're being very, cognizant of any sort of DNA cross-contamination. Not only is she using rubber gloves, but she's also using chopsticks, which we found was not a super efficient way to pick up guano pellets. Shocker. And then that middle picture.
I would disagree with that. Disagree.
The middle picture is kind of the end result. You throw these guano pellets in the solution, you label them, and then we ship them off to labs that have the ability to make this determination of whose poo is this. But yeah, kind of exciting project. Definitely a lot of fun. Turns out you need a lot of advanced degrees to rummage around under bridges and pick up bat poo. Who knew? So with that, I will pass it over to Chris.
All right, can someone verbally give me a confirmation that you can see my slides?
You got it.
Yep.
All right, thanks everybody. So yeah, last but not least, we're gonna kind of continue with the kind of environmental DNA theme. Sorry, I'm in a room that the lights turn off in. I figured that would happen. It may happen again. And so, but instead of talking about bats, we're going to talk about a set of semi-aquatic or aquatic species, in particular, 2 of our rare and declining turtle species. And so These two turtle species, one of which is called the Blandings turtle, the other is called the wood turtle. They are both currently listed by our state natural resource agency, so the state DNR, as state threatened, and both are being evaluated for listing at the federal level because their decline and their rarity is not only an issue within Minnesota, but it's an issue range-wide for both of these species. The Blandings turtle in particular is kind of an interesting and complicated species because it is actually quite widespread. You can see here in the map on the slide that there are quite a number of Minnesota counties that have records of this species, but that is a little bit misleading because it would maybe make you think, well, they're common, they're everywhere, but many of these populations are not doing well and are actually dominated by very old individuals. And these turtles can live over 100 years. And so just because there maybe are a lot of 50 to 80-year-old turtles in the population doesn't mean that the population as a whole is healthy, because you need those younger generations of turtles to have a healthy population. Wood turtles, on the other hand, are much more restricted, both in their kind of geography within our state and also within the habitats that they use. So they are almost exclusively a river and stream turtle, whereas the Blandings turtles will use a very wide variety of habitats, ranging from small wetlands, barely, more than, a knee's depth, very, very shallow wetlands, no fish, all the way up to the Mississippi River. We've seen Blandings turtles use the main big rivers in the state, and then kind of everything in between, from, big lakes and ponds to stormwater ponds even. And so some biological differences here. For those of you who have been involved on a project where you had one or both of these rare turtles, especially in one of the kind of more important populations, you know that avoiding, turtle impacts can be quite challenging, especially for a construction project, because in a lot of cases we're, digging up soil, which can actually be an attractant to these turtles. And so that makes it difficult for us to achieve our transportation objectives, while also trying to work with our colleagues in the natural resource agencies to, again, avoid and minimize those impacts. And this often results in conversations surrounding mitigation. So how do we kind of offset impacts that we have during these projects? And that mitigation can be very expensive, as it often is in the form of underpasses and fencing, similar to the discussion with the deer earlier. And many, again, kind of alluding to their long lifespan, many of the observations we have for these turtles are fairly dated. But because they can live so long, an observation from 50 years ago is still considered a current observation. And there just has not been the ability to more frequently survey populations to ensure that, when a record pops up on a project, that the population is still there. And it's not just kind of an old record and the population has declined and disappeared and really no longer would need to be considered for a project. And the reason that we haven't done these surveys more frequently is that they are very time expensive, time intensive, and therefore expensive. So the goal here was to use a DNA approach similar to the bat study that was just discussed by Ryan, but instead of collecting guano or feces in the environment kind of directly, we're actually collecting water samples, because these animals and organisms in general, and us included, we're always shedding our DNA through skin flakes and our waste products and even our breath. And so being that these two turtle species are closely associated with these wetland and water environments, we wanted to kind of further some of the research that has already occurred elsewhere and see if we can't get to a point where this is a viable tool for use for us as a transportation agency. And again, much like the bat example, we need to come up with a protocol that we can defend to the regulators, such that if we say, look, we collected samples and the results suggest that there are no turtles in this wetland, that they are, they're bought into the process to the level that then we can forego some of those avoidance and or mitigation discussions as a result. And so this project is kind of in between where the other two projects are. It is not quite complete, but it's in the home stretch. And so we do have some of that information to share, and then a few things that will be finalized here in the near future. But the overarching goal was really to kind of maximize our ability to detect the species while also minimizing costs. Can we do it quicker and faster at a lower cost ultimately? Vet these methods in the field so that, we're not just saying, well, it works in the laboratory, because the field adds lots of complexities. And then again, like Ryan mentioned, kind of get at how many samples do we really need to collect to have some level of confidence that when we say the turtle wasn't detected, that we can be confident that it's really not there. And then, getting at the kind of cost benefit, in what situation traditional methods, or maybe a combination of multiple tools, be the more logical choice. And so, again, from a kind of method standpoint, this is a fairly rapid process. You go out, you collect water samples with a number of replicates and controls, and you're seeing here, this is actually the research team out in a river up near Trunk Highway 23 and D1, where they're collecting some water samples. I got to tag along and document a few things. Turtles in this environment, again, are moving around in and out of water. They're feeding, they're, excreting waste. They're, doing a whole variety of their normal biological processes. And so they're shedding these DNA fragments into the environment. Those fragments can be larger and smaller based on how long they've been in the environment. And so we do have some degree of confidence that if we're detecting the species, that the fragments are sufficiently large, that the species was actually there pretty recently, because it degrades and breaks apart fairly rapidly. So we're not detecting turtles that maybe were there 5 years ago, for example. There is then some kind of water processing steps that has to occur. And then again, like Ryan alluded to, there's this laboratory piece. So those water samples and kind of the samples, after they go through all these filtering processes, are taken back to the lab. They amplify the DNA, extract it, and then send it off for sequencing to determine, well, what species, if any, are being detected. And what we found is that this does work. There is pretty significant seasonal variation in detectability. So depending on when those water samples are collected, you may need to collect many, many more or many fewer to have that relatively high degree of confidence that the species is absent from a site. And that's what these tables are showing here. The per sample cost is about $80 per sample, and that includes the actual field component and the lab work. And then when you kind of put it all together and say, okay, well, we know we have to collect multiple samples. What sort of cost would it actually take to, kind of do what we would call like a full survey using this technique? And if you collect your water samples in the fall, when it turns out is going to be the cheapest, where we have that highest detection, it may be as cheap as maybe $500, per pond or per stream. Again, it's going to depend on the number of water bodies in question. But if you collect water samples in the wintertime, which there might be some reasons why we want to do that, because you can drill through the ice and get into areas that maybe would require a boat or something. Unfortunately, that is when the detection of these species with this technique is the lowest. And so it's not to say that we can't use it in the wintertime, but it requires many more samples, and so it will cost more to do. So you're kind of seeing here maybe on the order of $500 to $10,000, depending on the time of year. But to put that into a little bit of perspective, some of those traditional survey methods that include a lot of staff time or contractor time, this still, even at the higher end, is likely cost effective, at least in some situations. And certainly at the lower end, this is likely a very cost effective way at getting at probable absence of these species ahead of jobs. One thing that's worth noting here is that our southeast population, so if you think back to that map of the wood turtle in particular, there's kind of a set of areas in northeast Minnesota and a set of areas in southeast Minnesota. The southeast Minnesota populations really do not appear to be doing very well. We had very few detections, and so there's more research that's needed. We did have detections. The laboratory methods were capable of detecting the DNA from those populations. So there wasn't anything like unique that made it undetectable, like kind of on the laboratory side. But it may not be a viable tool, at least yet, without additional research in those southeast populations. And again, kind of framing some of the cost perspective, and some of you may have, again, worked on projects that had turtle avoidance or mitigation. A single turtle kind of crossing with a culvert and fencing on a pretty narrow 2-lane road, starts, I would say, and this is probably maybe a fairly low end starting price at about $100,000. It can go up pretty considerably, pretty quickly if you need multiple crossing structures to meet some regulatory requirement. And so having that ability, to the tune of maybe a few $1,000s to rule out the need for some of those measures because the population is maybe no longer persistent, really could pay dividends in the long term for project budgets and schedules. And so similar to the bat study, I think this is likely to become an important tool in the toolbox. It's one of the many tools we have at our disposal, but again, trying to find solutions that allow us to perhaps do some of this work across a larger time period. So we're not, in this case, I didn't get to this, but for these turtles, a lot of the traditional survey work has to happen in really narrow time periods in the spring. Maybe it all has to happen within a 2-week time period during spring emergence. And so if you have a lot of sites needing this sort of work, it becomes a staffing impossibility to get to all those sites in such a narrow amount of time. And so this kind of gives some added flexibility with that as well. And with that, I think we are happy to take some questions.
Awesome. Thank you, Chris, Ryan, and Jesse. I always find your work so fascinating. We've got a couple of minutes left for questions, so feel free to put them in the chat. If you prefer to raise your hand and unmute and ask your question directly, you're welcome to do that as well. And following up on our question from earlier on what animal is National Appreciation Day today, the answer is bats. So if you put bats in the chat, good on you. I saw Joe's hand was up. Do you have a question, Joe? Oh, cannot unmute. Okay, sorry about that, Joe. I'm going to see if I can get that setting adjusted. Teams webinar is just trying to keep us nice and quiet on here. I'll see if I can adjust that setting for you. Okay, he'll drop it in the chat. We did have a couple of questions come in so far that some of our presenters have already responded to. So the first one, after the deer collision study, asked, were roadside mowing practices factored in? And Chris responded that it was not primarily because the data was not readily available statewide, but it could be factored into future models. We had another comment recommending the book Crossings by Ben Goldfarb. Another question on the deer collision study is who pays and maintains the fencing for deer crossings? And if our presenters, if any of you want to just chime in instead of me reading your responses, go for it. Chris said, it depends if required for transportation project mitigation or identified as a safety priority by the transportation agency. The DOT pays for it and maintains it. There are grants. In other situations, adjacent landowners or natural resource agencies may help pay for and or maintain. Fencing. DOTs typically pay for and maintain row fencing, and large animal fencing can be interpreted as another type of row fencing. For our bat detection project, we had a question come in on what are you seeing as far as culverts? And Ryan said, we occasionally see bat guano in large box culverts, but fairly rare. Vast majority of use is on span bridges. Smaller culverts are generally not considered suitable for bats, but there's a little bit of nuance there. And I'm seeing Joe's question in the chat for the turtle DNA testing. Is that a technique for turtles or a general use DNA test?
Yeah, so eDNA is super fascinating, and eDNA just means environmental DNA. So it's DNA left behind, and people are using it for a whole slew of purposes. We're probably most familiar, and you didn't even realize it, but departments of health were using it during the COVID pandemic, right? They were testing wastewater for the COVID virus DNA to get at rates in the general population. Within the kind of the wildlife side of things, people are using it to detect invasive species like the invasive carp that are moving into rivers and things. And then most recently, I've even seen studies that are actually being able to take air samples from the environment, and they're picking up DNA from air samples so they can tell what sort of wildlife are using the airspace in an area, which is just like super fascinating. But the more species you add to the analysis, there's more costs that go into it. And so typically when it's focused on, whatever the species, whether it's one or a few of most interest. And so in the turtle project, we're really focusing just on those 2 turtle species.
Thanks, Chris. We've got a question from Beth asking, can you explain a little more about the variation in eDNA sampling costs in the fall versus winter?
Yeah, so it all comes down really to the amount of water samples that need to be collected, processed, and processed through the laboratory. And so in the wintertime, and we talked a lot about why this may be, but in the wintertime, I think we're leaning towards the hypothesis that, well, turtles are sleeping, they're at the bottom of, lakes and streams and things. They're not, moving around, they're not shedding as many skin cells, and they're not, excreting waste products and things as much. And so there's just less DNA floating around in the water sample during the winter months. And so therefore, if we want to have a 95% confidence that we're adequately sampling that site to determine absence, you have to collect just a ton more water samples, which adds cost.
Thanks for explaining that difference. Got a question from Nathan asking, could these water DNA tests, like for turtles, be used for super accurate lake fish surveys or other multi-species surveys?
Yeah, and this is getting a little bit beyond my area of expertise. And I know we have a couple genetics people on the listening in. And so thinking, Tricia, if you maybe can answer whether or not some of these techniques can actually like quantify abundance, because I'm not sure. But what we're doing right now with the eDNA is we're just getting at presence absence. So we really can't say much about, well, are there a lot of these rare turtles, or is there just one and we're detecting it? And so a lot of the fish surveys that, like DNR does to set like fish, fishing limits and things of that nature, they need that abundance data. So they do those netting surveys, and they look at like how many individuals of different size classes, and then they, can prescribe their management recommendations based on that. And I'm just not sure if this technology is quite there yet. But if it's not, I suspect it will get there. And so it may very well be used for that sort of thing in the future. But somebody else, if you're on and you know more about the ability of eDNA for quantifying abundance, please chime in the chat.
Hey, Chris, it's Trisha here. If you can hear me.
Yep.
I don't know, I guess for this question about abundance, I'm not entirely sure on where eDNA stands, but I will say that the primers, the genetics that are looking at those specific segments of DNA for any given species have to be developed for a particular species. So it's not like you can just take a water sample and be like, oh, let's, look for X, Y, and Z. You need to bring that back to the lab and then develop those primers, which usually, has an added cost and takes many weeks or months. And some species, including turtles, I think were a little bit trickier than maybe some others. I know they've been doing this with fish, with salamanders, with all sorts of crayfish, all sorts of other animals. It's pretty amazing technology now. But yeah, they have to develop those, and we're still kind of at the very beginning of what all we can do with this.
Okay, thanks, Trisha, for chiming in.
Thanks, Trisha. Okay, I'm not seeing any other questions in the chat. So thank you all for joining today. We'll send a follow-up email with all of the references we mentioned, and then also the links to the project pages. So the deer collision study is already complete. You can access that full report in the research summary. The other 2 studies are in progress. We do have the option to subscribe to the research project, so we'll send you an email when the project is complete. I don't expect you to be coming back to our research project database to check in on that. And if you enjoyed today's session and want to keep learning more about transportation research topics, we encourage you to attend our upcoming Research Radar webinar series. So thank you all for coming, and another thank you to all of our wonderful presenters today.